So the topic of the lecture today will be the modification of radiation response. We have now seen the basics of radiation action. Now we see how this radiation response can be modified by physical and chemical factors. So the lecture will be divided. It's a pretty long lecture. It will be divided in 6 parts. We will first talk about the intrinsic, the genetic radiosensitivity. Then we will talk about fractionation and dose rate effects, cell cycle dependence, oxygen effects. We already saw something in chemistry here. Radioprotectors and radiosensitizers, also here we saw something in chemistry. And finally, all of these effects, we will see what happened when we move from X-rays to highly heavy ions. So first, we have to start with the definition of the 4R of radiation biology that are extremely important in radiotherapy. The 4R justify the use of fractionation in radiotherapy based on the following factors. Repair of cellular damage is the 4R, so the fact that you can repair the damage, that this we have seen in the DNA and chromosome studies. Reoxygenation of the tumor, we have seen something in the chemistry part, but this essentially means that if you fractionate the dose, we will see the tumor can be reoxygenated and then becomes more sensitive. Redistribution within the cell cycle, which is now the cell cycle dependence, and repopulation of the cells. So you see that to understand this 4R, we need the lecture today because, you know, repair includes intrinsic radiosensitivity, reoxygenation is the OER, the oxygen enhancement ratio. Redistribution within the cell cycle is the cell cycle dependence. Repopulation of the cells is a tissue effect. We will not touch it strongly here, but simply means that you can repopulate your tissue if you kill many cells. However, the 4R were now are more than 4. There is this is also how you can represent the 4R, let's say from the more from the practical point of view. We will see why we have this curve. What is reoxygenation? Repopulation, I told you, only means that you can repopulate a tissue that has been severely damaged. There is a 5th R, which is the intrinsic genetic radiosensitivity. So independently from everything, different tumors and different normal tissues, you see here, these are 80 cells, ataxia-telangiectasia, very sensitive, bone marrow cells, mammary cells, jejunal cryptal cells, thyroid, they have different sensitivity to radiation. So this genetic radiosensitivity is the 5th R of the radiation. And now there is even a 6th R. There is a recent paper where, in addition to repopulation, repair, redistribution, intrinsic radiosensitivity, and reoxygenation, we have reactivation of the immune response. So that's still another R that is related to the immune response to radiation therapy. This is really more in radiation therapy. We will stick in this lecture to radiation biology points, but that's just to let you know. So the first then is the radiation, is the genetic sensitivity. That's the first topic of today. So radiation protection standards assume that the human population is uniform in radiosensitivity. Now, this is a problem. If there is a radiosensitive subpopulation, this will distort the shape of the dose-response relationship and make it unethical to put such individuals in the area of high dose. It is, of course, extremely important also in radiation therapy because, you see, this is now the NTCP, the normal tissue complication probability, which is really what limits the radiation response. You see, N is the average normal tissue complication probability. With generally, the limit is 5%. You don't want to have more than 5%. So the dose will be fixed by this curve. But actually, you see, if you have a sensitive population or even a hypersensitive population like the AT, clearly this dose will be so high, too high. And if there is a resistant subpopulation, you see more resistance than the normal, which is unclear, well, then clearly you can go up in dose without affecting the normal tissue complication probability more than 5%. So there is a huge impact in radiotherapy, and there is a huge impact in radiation protection. What is how can we have radiosensitive or radioresistant individuals? Let's say generally there are three categories. You can have mutations in the so-called high penetrance, low frequency genes, mutations in low penetrance, high frequency genes, and SNPs. SNPs is single nucleotide polymorphism. So when you have mutation at the levels of the single nucleotide, we generally do not have clear phenotypic effects. So you cannot tell that there is a given mutation in that individual. SNPs are very common among the different individuals, but these SNPs are those that can then play a role in radiosensitivity. Of course, if you take the high penetrance, low frequency genes, the high penetrance, low frequency genes are genes like ATM, that is, of course, essential for DNA repair. BRCA1 and BRCA2 and RAD9, these are genes also involved in homologous recombination, and the BRCA family is well known to be involved in breast cancer. It's called BRCA for breast cancer. This is, you see how this kind of genes, you know, this is ATM, CTIP. We have seen also CTIP that is involved in resection, BRCA1, RAD9. These are genes that can, are, let's say, easy candidates to say, okay, if you have a mutation in this gene, probably you become more radiosensitive. This is already well known for ATM. In that case, however, is a very phenotypically obvious effect. You have clearly a difference in AT patients. Now, this is clear also when you have breast cancer and chest X-rays. This is an old study from almost 15 years ago, about 15 years ago. This was women exposed to chest X-rays, and clearly, and this was in BRCA1 2 mutation carrier women. And you see the hazard ratio. The hazard ratio is essentially the ratio of cases of breast cancer in the test population divided by the cases of breast cancer in the control population, is 1.54. This is the confidence interval in the whole cohort. And if you take a very young aged exposure, is almost 5. So it's, you see, going down with aged exposure, this becomes extremely high. Of course, these are cases with women that had 1 to 5 X-rays, but the dose is about 0.6 milligray, so it's very low. It's extremely low. But this notwithstanding, you see this BRCA1/2 carrier are very sensitive, very sensitive. Now, there is another cohort that is very well known and is particularly interesting, and is the cohort of meningioma in Israel. This is a meningioma. This is a tinea capitis. Tinea capitis is a worm, is a ringworm of the scalp. It's an infection that makes this plaque. And after World War II, there were thousands of children that coming back to Israel, or going to Israel to be precise, and they were irradiated with fairly high doses, like 1 to 6 gray of X-rays, to get rid of the tinea capitis. Sure enough, you had an increase in meningioma. Meningioma is a well-known brain tumor. It's benign in nature, so it's not a malignant tumor, but still is quite, in many cases, has to be removed because it's too big. Overall, there was a 1% of the irradiated children developed meningioma. So again, you see, even if the dose was fairly high, the fraction was relatively low. But what is particularly interesting is that the increased risk is basically caused by a subgroup of 17 families out of 160, where the risk in these families was 4 in 5. Keep in mind that these are generally families, these are fundamentalist Jewish, they have many children. So in the family, surprisingly, you had like 4 out of 5 children with meningioma, only in 10% of the families. In the other families, you had 0. So you see, 1% of the control families had multiple cases of meningioma in the same family. So if you take the controls, it's a low frequency of multiple cases of meningioma. So how do we explain it? Clearly, it's a strong point showing that there is a genetic component in the sensitivity to cancer. And there was a SNP analysis that suggests co-inheritance of multiple risk alleles rather than a single gene. But this really begs the question of whether radiation carcinogenesis is only a matter of a few, not of everybody. Maybe for us, for many of us, radiation carcinogenesis is not an issue, but for few of us, it's very dangerous. So the data from tinea capitis now, the more modern techniques, let's say, for this kind of analysis is the GWAS, genome-wide association studies. Generally in the GWAS, you have this kind of plots. You see, these are called Manhattan plots. So you see all the chromosomes are here, and the bands within the chromosomes, you see this is the different location within the chromosomes. And this is the probability of association. It's a sort of correlation coefficient, if you want. So when you have this, let's say every point gives you the SNP, and you see that in this specific case, where there's nothing to do with radiation, is a case of constriction, a small blood vessel. Is a study on microcirculation. You see that constriction in small blood vessel seems to be associated with SNPs. You see quite significantly with SNPs in chromosome 6, chromosome 12, and in chromosome 19. So this single nucleotide polyphosphate. Can we do this in radiation studies? This has been done recently in patients treated with radiation when they were children, so in pediatric tumors. In particular, this is a study on Hodgkin's lymphoma, where essentially you have to irradiate the chest. So you also irradiate the breast. You can give a very high dose to the breast, 10 or higher gray breast radiation exposure. And then you look at the correlation between SNPs and development of breast cancer in this patient. And in particular, you see there is this is the map of the chromosome 1. There is a clear SNP here. This is called, using this kind of mapping, is called RS4342082. Don't ask me. Somehow is a way to design the map of the chromosome. This SNP is actually close to, not really on, but close to a gene which is called PROX1. This is a transcription factor. So it's involved in early embryonic development and implicated in cellular proliferation and migration. So clearly is a good candidate for being involved in the development of radiation-induced breast cancer. So these kind of studies are now becoming more and more popular. There's a radiogenomics cohort, and is important both, like in this case, for the induction of cancer to understand the sensitivity to stochastic effects, but also for acute effects for radiotherapy patients, because we would like to know before the therapy what is the genetic sensitivity of the population. Now, the second point that we will touch are the dose rate effects. And in particular, we will see the so-called sparing effect and the DDREF, which is the dose and dose rate effectiveness factor. I think we mentioned this already in the chemistry. So this is an old idea, but I think it's important that you are aware of it. The idea is that there are 3 types of radiation damage. One is the lethal damage, which is irreversible and leads to cell death. Then you have the sublethal damage. The sublethal damage is the one, let's say is the one that we use in the linear quadratic model. No, so you can have a lesion. This lesion can be repaired, but if 2 lesions interact, either intra-tract or intertrack, so alpha D plus beta D square, then you switch into lethal damage. Now, the sublethal damage repair is what actually causes the sparing effect, because essentially the probability of having intertrack, so the beta term, the probability of having intertrack event will be lower if you reduce the dose rate, because now if the time of repair becomes of the same order of magnitude or even shorter than the time of irradiation, then of course you can do a lot of repair before you have a second event. So in the linear quadratic model, repair of sublethal damage means that you should switch from a linear quadratic to a linear term. There is a third type of damage, which is called potentially lethal damage. This is not strictly related to the alpha-beta model. It only means that you can modify, if you change the environment, you can modify the lethality of some lesions. This is typically seen, you see, in the potentially lethal damage. This is typically seen in the so-called delayed plating experiment. So if you irradiate cells that are in resting state, they are confluent. You irradiate them, then you replate them after irradiation. If after irradiation you keep them in resting state for a long time, then you have a lower survival. So this is an example. You see delayed plating, and this is an old experiment from 1973. But you see here that if you keep the cell 0 hours, if you irradiate the cells and immediately after you replate them and you look at the colonies, you have this survival curve. But if you wait 6 to 12 hours and then you plate them, the survival curve goes up. So you have a higher survival. This is called repair of potentially lethal damage. Is this also observed in vivo? Somehow, yes. You see here in this case. In this case, you have tumors that are irradiated in vivo, in mouse, of course. And then the tumors is taken out and is replated. So this is the time between irradiation and explant. And this is the relative survival. So you see is that if you increase the time, the survival goes up. And this seems to be even more significant if you take large solid tumors than small solid tumors, maybe indicating the possible relationship to the hypoxia of the tumors. I will see later. But as I said, sublethal damage repair is really what determines the dose rate effect. Now, if you think to the linear quadratic model, to the microdosimetry model, if you want, I mean, the idea is that you have alpha, beta. Alpha is intratrack, beta is intertrack. Now, if you go with beta, if beta goes to 0, your linear quadratic curve will become more and more linear. There must be a point where this cannot be longer increased. I mean, so there must be a minimum. There must be a point at which an increase in the number of fractions or a lowering of dose rate will no longer increase survival. So there must be a limiting dose rate, let's say, because once you have perfectly linear, it should not go down anymore. This is actually seen. You see, this is also an old experiment, Mortimer Elkind, 1965. Actually, this shows the dependence on the time between two doses. So you wait, you see, if you wait for only 1 hour, that's not really the maximum increase in survival that you can have, because if you wait longer, you reach a plateau. This plateau shows, you know, that now you have completed the repair here before having a second hit. And this is in fact how it looks like. You see, now this is not fractionation. This is only changing the dose rate. So this is a survival curve here at very high dose, I mean, conventional dose rate, 107 rad per minute, which means 1 gray per minute, effectively. And then you reduce the dose rate. And then you see that reducing the dose rate, the survival goes up, and, which is particularly interesting, you see the curvature is reduced. So the dose response curve has a tendency to become linear when you go to low dose rate. This is called sparing effect. So the impact of radiation at low dose rate, say the general assumption is not always true as usual in science, but the way you have to think is that when you have low dose rate, you expect a lower effect. This is true for survival, of course, but it's also true for chromosomal aberrations here. So this is a typical dose response curve in non-cycling human fibroblasts. These are actually asymmetrical aberration per cell in GIMSA, so it's dicentrics plus rings, nicely linear quadratic. If you go down in dose rate, it becomes linear, and please note, this is interesting, 0.1 centigrade per minute or 0.046, so 1/2 of this centigrade per minute, gives the same result. So these are the triangles up and down, meaning that when you are around 0.1 centigrade per minute, you are in the region of the limiting low dose rate. So going down again in dose rate is not changing the situation, which makes sense in the linear quadratic model, because now you have only intra-track events. And if you have only intra-track events, the dose rate doesn't matter. It's a little bit different, but here is probably different to see in principle, this means that if you have a linear dose response, you should not see a difference. These are also all data on neoplastic transformation. Neoplastic transformation also showing a clear sparing effect. You see 100 rad per minute, 0.1 rad per minute is going down. Of course, these data on neoplastic transformation are particularly important because this means that the cancer risk, the stochastic risk, this we will see in humans in our exposure, should be considered reduced when we compare our risk to cohorts of people exposed acutely at high dose rate, like Hiroshima and Nagasaki. And this we will see in the other lecture on deterministic and stochastic effects. And you see, it's also interesting that the R of radiation are entangled somehow. This is an experiment on human lymphocytes taken from different individuals and irradiated either at high dose rate or at low dose rate. Interestingly, you see, when you irradiate at high dose rate, you have a variability, but if you look at the real points, this variability is almost within the uncertainties, the variance of the different curves. When you go to low dose rate, you see better the difference. So somehow the irradiation at low dose rate enhance the differences in intrinsic radiosensitivity. And this is probably because, you know, when you go to intra-track damage, then the only things that matter is how good the cells can repair the damage. So the intrinsic radiosensitivity becomes more clear when you go to low dose rate rather than high dose rate. You see, this is over 40 human cell lines irradiated. Many factors contribute to dose rate effects. I told you already that the cells with a broad shoulder in cell survival will be less susceptible to low dose rate. The duration of the cell cycle we will see is important because there are radiosensitive and radioresistant, and the ability of the tissue to adapt. So I'm sorry, now that I look at it, this is cellular sensitivity of stem cells. So cells with the broad shoulder in cell survival curve are less, it's not that are less susceptible to, so are actually, if you talk about susceptibility, I should write, I apologize for this, they're actually more susceptible. Sorry, I realized that there was a mistake. I don't know where did I take these slides, but if you think about the shoulder, I mean, if there is no shoulder, then there is almost no difference when you change the dose rate. There can be no difference because you are already in the alpha. So it is when you have a broad shoulder that you have a dose rate effect. So I apologize, it should be more. I'll change it also online. Now, in terms of radioprotection, how do we represent it? Generally, the way we represent it is the so-called DD-ref. DD-ref is here, goes directly in the definition of the risk as a function of the dose. So the risk as a function of the dose, as this term, which is a term essentially that takes into account the quality factor times the dose. So this is a dose, if you want, is a dose equivalent. So it's a dose in sievert. You multiply this dose for a risk coefficient that comes from epidemiology, but then because epidemiology is generally high dose rate and high doses, you have to divide for a dose and dose rate effectiveness factor, which is broadly defined as the alpha of the high dose rate divided by the alpha of the low dose rate. So what happens is that the shape of the DD-ref is essentially this one, is 2 when you go to low dose rate, and then goes to 1 when you go to high dose rate. Actually, rather than 2, there are more studies now showing that, for example, the BIR7 in the United States recommends 1.5 and not 2. Actually, this factor is a very large uncertainty. It's one of the main sources, we will see, main sources of uncertainty in the estimate of risk. And in addition, but this we have already seen in the chemistry part, the uncertainty is not only in the low dose rate region, but also in the very high dose rate region where apparently you have this flash effect. We have seen this in the chemistry lecture, where you have protection when you go to very high dose rate. This still has to be clarified, this phenomenon. Low dose rate and fractionation are, of course, the same, is not the same thing, but it's the same mechanism. So essentially, if you irradiate with the dose D, you have this survival, but if you split the dose in 2, you will get a higher survival. It's like low dose rate, if you want, because of course here, if you wait enough, you remember these old experiments of Mort Elkine. You see, you have to wait several hours to be sure that you have full recover. And then at that point, you will have this kind of effect. And of course, if you fractionate many times, you get a better survival. And that's what you do to spare the normal tissue during radiotherapy. We will see that this is not true anymore, but traditionally it was said if you shoot in one spot, you kill the patient. You have to fractionate. Now, in the linear quadratic formalism, how do you represent this effect? Well, it's easy if you think about if the effect is alpha d plus beta d square. When you have n fractions for a total dose of nd, then you have to write n alpha d plus beta d square, that you can write in this way. And if you have many fractions, so that this factor goes to 0, then the ratio of the single fraction divided by n fraction becomes 1 plus beta over alpha times d. So you see the alpha beta ratio, how important is the alpha beta ratio? Because you see that if the alpha beta ratio is extremely high, or the alpha beta ratio is extremely low, the situation will be completely different, you see. If alpha beta is very high, beta over alpha goes to 0, and then there is no difference between E1 and En. And in fact, if alpha beta is very high, this means that beta is very small. This means that the survival curve is linear, and then there is no effect of fractionation. This we have seen already. No sparing effect, no effect of fractionation. The other way around, if alpha beta is very low, so you have a broad shoulder, beta is very big, then there will be a big difference, a big reduction, if you want, in the effect if you fractionate n times. This is all in the alpha beta ratio. So the alpha beta ratio gives you is the most important radiosensitive parameter. If you have the alpha beta ratio, as you do have from clinical practice in the clinics, then you can estimate the impact of fractionation. In another way to represent it, we can put it in this way. You see, if you fractionate now, if you look at the survival curves, then the number of survivors would be N0, E2 minus alpha D plus beta D square is the first one, then times E2 minus alpha D plus beta D square, the second one, and so forth for N times. So the total survival after N fraction can be written in this way. And this is simply this quantity, D times 1 plus D over alpha beta is called BD, biological effective dose. And then you can say that the survival is simply E2 minus alpha BD. So you see that from the alpha value and the beta value, you can estimate what will be the impact of fractionation, what will be the impact of having N times D. D is the total dose, so D, capital D is N times small d. So third will be the cell cycle dependence. Now the cell cycle we have seen it already. This is only to remind you that we have G1, where essentially the cell grows until the checkpoint. Then it duplicates the DNA in the S phase. Then you enter another gap phase, the G2, with a further checkpoint here before entering in mitosis, and then you have two cells. Now, the sensitivity in the different phases is different. So if you irradiate the cells in different phases, you get different survival curves. The same cells, and quite significantly, you see, this is N phase. If you irradiate them in N phase, or let's say in late G2, this is G1, and this is early S phase and late S phase. This is only the a magnification, if you want, of the mitosis. This is the mitosis times 2.5 to give you an idea of how big is the effect, the difference between late S and G2 and G2M. So S is radioresistant, G2 is radiosensitive. And in fact, if you irradiate the cells, let's say these are CHO cells, these are difficult, this is a very old experiment, again, from 1966. Essentially, you collect mitotic cells by shake-off, because they are rounded. So if you shake the slide, you collect mostly mitotic cells. Then you irradiate them at different times. Of course, you plate them in different flasks, and then you irradiate every flask at different time. Of course, you lose synchronization, but approximately, you see, you start here with cells that are in G1, you see at 1 hour after shaking. Then you are in S, and you see that the surviving increase very much, S phase. Then they enter G2 and the survival decrease again until in M is now back to the lowest value and then goes up again. So this kind of behavior is common. You see, this is again other experiment, and this is in Chinese hamster cells, V79, that have a very short G1, you see here. So this is M, G1, S, where you get radioresistance, and then G2M. And this is instead in HeLa cells that have a longer G1. So if they have a longer G1, you see, you don't see it here, but you see it here, that during G1, you also go from high sensitivity here to a lower sensitivity here. Sorry, from more resistant. In M is very low, then increase in G1, then decrease a little bit again, very resistant in S phase, and then down again. The problem of the cell cycle sensitivity is important for what is called the reassortment of the cell. So essentially, and this is why also fractionation is so important. So when you make a split dose experiment, you will have a preference for killing cells this is now the cell surviving fraction as a function of the time between the dose, and this is the repair of sublethal damage. So if you want, is the sparing effect. You see that if you wait 2 hours, you have increase, but then you have reassortment. This means that at a certain point, you kill the cells in the most sensitive phase, and you leave the cells in S phase, essentially still alive. So your surviving fraction goes down until you start to have repopulation. Then it brings you up again. This is what we want to do in radiotherapy too. We want, if you give only one fraction or a few fractions, there is a risk that you leave the more radioresistant cells in S phase alive. And then to correct this, you want to fractionate so that eventually you will kill, you will reassort, you have a reassortment in the cell cycle, so that cells that were in S phase yesterday, tomorrow will be G2, so they will be sensitive and will be killed. This is the principle of the R in the 4R. One is reassortment, and that's why you have the reassortment is so important. What about transformation? So far we focused on cell killing. What about transformation? Well, this is again the cell surviving fraction. These are actually C3H21 alpha cells. Experiment in the Columbia University in New York. So these cells, you know, these are murine fibroblasts, very convenient to look at cell transformation. If you look at the cell killing, this is the time post-synchronization, again with the shaking effect. This is the value for asynchronous cells at 3 gray, and this is with the shaking. You see again the usual problem. You have, this is G1, then it goes down in S phase, and then when you go to G2, it goes down, sorry, this is G1. You have a decrease here, then during S phase, the survival goes up because resistance is higher, so the survival goes up. Then when you enter in G2M, goes down again. So this is the typical behavior. We have seen it approximately the same in the HeLa cells. You see, these were all the experiments in the HeLa cells. But surprisingly, when you look at transformation, well, it looks like, you can look also at neoplastic transformation. Now it looks like you see G1S, but then around the G2 here, G2 or G2M, you seem to have an increase, at least a factor of 2, if not more, in neoplastic transformation here. So it looks like there's a window of sensitivity to neoplastic transformation, which is in the G2M phase. This problem, you see, this goes in saying what is the dependence of the dose rate for transformation. There are some data showing an inverse dose rate effect. These are essentially data that come from neutrons. So we will come back to this point later. So the general conclusions for the cell cycle are that cells are most sensitive to radiation at or close to M. They are more resistant in late S phase. For prolonged G1, there is a resistant period in early G1, followed by a sensitive period in late G1 before entering in S phase. Cells are usually sensitive to radiation G2, almost as sensitive as in M. There are, of course, exceptions to these rules in some cell lines, but this is the general behavior. Oxygen effects. Now, oxygen effects, we have discussed this already in the chemistry lecture, how important is the oxygen, you know, for the fixation of the radicals and for the fixation of damage. And we also say that the more oxygenated are the cells, the more sensitive they are. And this is expressed in what we call oxygen enhancement ratio. So the oxygen enhancement ratio, or OER, is essentially the ratio of the dose in hypoxic and normoxic. Let's say, normally you assume something like, let's say, 0% and 20%, which are the extreme. They're not really like that in the normal tissues, but this would be the case. And this is known as oxygen enhancement ratio. So you see, this is an example. This is a survival curve, hypoxic and oxic. The OER here at the high dose rate is 3.5. If you go to low dose rate, here essentially you lose the curvature of the dose response curve, but you still have the OER. You see 2.5, a little bit reduced, but 2.5. If you use the alpha beta model, then the OER can be calculated, let's say, with this formula. This formula includes the alpha beta model. This we already saw in the chemistry, so I don't need to go back to this one. Only one thing is important, that hypoxia can be chronic and acute. So in most cases, it is acute. It is chronic hypoxia. So you are chronically in hypoxia. And this is also difficult to reproduce in lab experiments, because you have to keep yourselves for a very long time in all animals, or for a very long time in hypoxia. But there can also be acute hypoxia, where, and this is what is discussed also to explain, you remember the flash experiment, when essentially you can have occluded blood vessels that cause let's say, hypoxia acutely, quickly. From the point of view of the clinics, it is known that hypoxic tumor have a poor prognosis. This is a famous old experiment. This is the survival of uterus cancer, looking at small oxic tumors. These are small hypoxic tumor and large hypoxic tumors. You see, the survival gets much worse. But what is the dependence from the PO2? So we always say hypoxic, oxic, but be careful, because in reality, as I said, there is not really such a thing, because the 0% will be a dead tumor. So there's not really 0%. And 20% oxygenation is probably only in the blood, you know, when you have the hemoglobin, and the lung can be like 17%, you know, something like almost more than 15% is only in the lung. Most of the other tumors, about the other normal tissue, end of the tumors, are in an intermediate range. So how the radiosensitivity, the relative radiosensitivity here, depends on the oxygen tension. You see, if you start to 1, this is essentially the OER, if you want. If you start to 1, this is a typical Michaelis-Menten kinetics. It goes up and then plateau. So this is air, and this is 100% oxygen. This is the oxygen tension. And is in this region, at 3 millimeter, you have about 1/2. This is about 1/2. This is about 3. So it's quite sharp here. It's in this region, which is actually the most important region. You have a sharp increase, and then you have a plateau. This is seen now. This is the relative radiosensitivity. This is seen in terms of OER. So you have an OER that at low PO2 concentration is around 3, and then it goes to 1. This is 1 here when you go to high oxygen concentration. So essentially hypoxic, and you see hypoxic, but you see the region, this region here between 1% and 0.1% is a critical region, and that's a region where the OER changes sharply. This is very hypoxic. Almost never you have this case. And this is very oxic, almost, of course, 100% oxygen will be impossible. Let's say this means that you are in deep hyperventilation, but you can have some 10%, 15% in the lung, for example. But most normal tissues will be here, somewhere in the few percent region, and most tumors will be somewhere here. So this is extremely important to understand. There is some discussion on whether this OER, as you see here, is survival dependent or not, if you take the linear quadratic model. But in reality, most of the data seems to show that it is basically, you see, if you measure it at 10%, 50%, 1%, it is almost the same. So it is more like a dose modifying factor than an RB. RB is dose dependent. OER does not really change the shape of the curve, the OER. So this means that it's almost the same at different survival level or at a different dose. Now, what is the basic idea of the reoxygenation? So let's go back to the situation why tumors are hypoxic. Tumors are hypoxic essentially for this kind of problem, that the cells that are close to the capillary will be highly oxic. But then if you get away, you are more hypoxic. If you have a tumor, the tumor grows very quickly, so there is no way that the capillary can reach the center of the tumor when it is growing. So generally speaking, we will see that this is not true. I mean, this image, which is very well known, is not really true. We know today that there is not a situation where there is a center hypoxic and an external part which is nicely oxic. The situation is much more complicated. The hypoxia is much more diffused in the tumor. But let's assume that it's like that, for simplicity, just to understand the logic. Let's assume that you have oxic, here you can get the blood vessels, so this is nicely oxic, but then you go in depth and this becomes more and more hypoxic. So you have a hypoxic core and an oxic region outside. If you irradiate, you kill the oxic cells, but you don't kill the hypoxic core, but the OER can be as high as 3. So this core is now alive. But then you wait. If you wait 24 hours, then the blood will add oxygen here. So when you irradiate again, this will be the situation, and now you can kill again these oxic cells. This will be the hypoxic core, and so on and so forth. So the trick, let's say, of fractionation helps for the oxygenation, because as you see, the reoxygenation will increase the sensitivity of the tumor. Finally, the last point that also we mentioned in the chemistry part is the radioprotectors or radiosensitizers. Let's say radioprotectors and radiosensitizers are both useful in radiotherapy, because you want radiosensitizers for the tumors and radioprotectors for the normal tissue. Of course, in radiation protection, you only want radioprotectors. Radioprotectors are only that you want, and there can be different types, including not necessarily drugs. There can also be dietary supplements, and especially antioxidants, because we have seen that antioxidants reduce the reactive oxygen species, which are the most important to induce sensitivity. So they can be important as radioprotectors. So this is from Jim Joseph, who is a researcher from the United States. He said once that fruits and vegetables may become as important in space-going vessels for space radiation protection as limes were on the sea-going vessels of old. Generally speaking, the radioprotectors, one can assume, you can imagine that they work when you have indirect effect, no? Because of course they protect from the radicals. And for UV, a great part is indirect damage, very great part. So, and you see that impact of scavengers. So scavengers are those chemicals, you remember in the chemistry lecture, that will remove the free radicals. Is a very strong impact on the this is the DNA damage. In radiation biology, the one of the best scavenger used is the DMSO, the dimethyl sulfoxide. Dimethyl sulfoxide is very well known as an anti-freezing agent. It's used a lot, was used a lot in the engines of the old time, in the radiators, was added to the water. And you see here in this study, this is the survivance fraction as a function of the concentration of the DMSO. And you see nicely that this is 0 molar, And then if you increase, you go up and up here, you see that you have a increased survival. Now, if you define the increase the degree of protection with this formula, the logarithm of SF0 minus the logarithm of SFX divided by the logarithm of SF0, and you calculate it here, you have this kind of curve. You see, as a function of the DMSO concentration, this goes up, but does not really goes to 1, stops at something like 76%, 80%, which corresponds to the fact that when you use X-rays, 20% of the damage is still direct damage, is not indirect. So it's not mediated by free radicals, but it's direct damage in the DNA. However, you see a good 70, 80% of the damage is mediated by free radicals and then can be scavenged, can be protected. It's a very high fraction. But do we have drugs that can do it? Not really, not really. Actually, one of the few drugs that are used in the clinics is the so-called WR2721, also called anifosfine. So where is this coming from? Why this strange name? These are names that come from the time of the Cold War, when United States and Soviet Union were threatening each other to have a nuclear war. And then there was a lot of research, because nuclear means radiation exposure, a lot of research on products that were radioresistant. So there are many WR molecules. WR stands for Walter Reed Army Institute, because this was a research done in the Army. Actually, 2721 was one of the best, and is now FDA approved as radioprotectant, I'm sorry, not cryoprotectant, radioprotectant for clinical use in radiotherapy and chemotherapy. And this also, you see this an experiment instead on tumorigenesis. You see that control 60 centigrade plus WR is very protective also for tumorigenesis, but it's a molecule that has substantial side effects, including nausea, vomit, hypotension. So it's okay for cancer patients, you know, if they have like head and neck where you have severe side effects, you can use this one. But because in any case, you know, cancer patients are used to this kind of side effects. But if you take normal individuals, not to mention astronauts, that would be really, really dangerous. There are other molecules much more attractive. These include, for example, aspirin. Aspirin, this is a large experiment on aspirin, but also other molecules like Valium. The problem is that how effective they are in vivo is not so clear. These are in vitro experiments that seem to be very effective, but in vivo seems to be not so effective. On the sides of the radiosensitizers, one molecule that is very much studied now are these gold nanoparticles. They seem to, you see, this is the gold nanoparticles can go specifically into the tumors, because these are nanoparticles, so it's only nanometer size. They're only 2-3 nanometers in diameters. And you see that they can actually increase the sensitivity. The mechanism of the increase is probably due to the emission of Auger electrons from the gold, because now you have a high probability of Auger electron emissions for these high Z materials like gold, and these Auger electrons will have a strong effectiveness because they are low energy around these nanoparticles. Okay, in the final part of the lecture, we will now see all the effects that you have seen before, how they are changed if you use heavy ions, so higher lit in general. So first, the individual radiosensitivity. Now the question is, does the RBE depend on genetic sensitivity or not? Generally speaking, we will say that if you have high radiosensitivity, you should have low RBE. You remember that the RBE is also strongly dependent on the alpha-beta model. And of course, if you have a broad shoulder, you have a high RBE. But if you have already a linear dependence, you should go to an RB which is approximately 1. Now, but the question is whether you have a different genetic response. So maybe if is it true that if I'm radiosensitive to X-rays, I'm also radiosensitive to heavy ions or whatever? Maybe not. You see, this is a nice experiment that shows these are genes that are, I'm sorry, the reference is missing here. I think you should see it in the file. No, it's been deleted somehow. It is, these are genes that are expressed at least 2 times higher after irradiation to carbon or neon in comparison to X-rays. And these maps, these are maps that are built at the computer from which genes are correlated to those genes that are activated differentially, you see. And this is the kind of genes. So there is a number of genes starting from TP53, this is P53, that are activated in a differential way when you use, this is MYC, and this is CBRA, which is a transcription factor. These are also, all of them are transcription factor, but you see there are other genes that are dependent from them. And this is instead another paper showing, now these are cells, so human bronchial epithelial cells that were irradiated with 3 different types of radiation: gamma rays, low LET, silicon, intermediate LET, iron, high LET. And they looked to the number of genes that were expressed after irradiation. And you see that some genes were expressed after all kinds of irradiation, 67 to be precise, a very large panel. But some were really unique. You see, 173 were expressed only after gamma rays, 191 only after silicon, and 107 only after iron. So there is room to believe that the genetic sensitivity can be radiation quantity dependent. The dose rate effect, now again, if you go to beta 0, you should have no sparing effect. So in principle, because there should be no dose rate effect, because we say that the dose rate effect is all about eliminating the intertrack effect. And in fact, I mean, these are experiments with fractionation. Some old experiments even show possible potentiation if you fraction it, but this is very heavy ions and very low energy. In most cases, you see, like in this case, this is the this is the ratio of the inactivation as a function of the time after irradiation, and you see here that the data are approximate. This is argon and krypton, so that you have a tendency to have a reduced dependency on the time between irradiations when you go to high LET. So generally speaking, that's how it should look like. No, if you have acute dose X-rays, you should have a perfect linear quadratic that when you go at the maximum sparing effect, this should become linear. So this is what should happen. Now, if you already have this situation, if you are already high LET, heavy ions, high RB, you see here, what will happen? You see, when you have alpha particles here, not X-rays, but alpha particles acute, well, in that case, you do expect that this is not changing in low dose rate because there is no intertrack effect. So no curvature, no dose rate effect. This is the dogma. It's not always true, but makes sense. You see, somehow this is again the chromosomal aberration now with alpha particles. These are the total aberrations, and you see they are linear. Dicentrics also linear behavior, and there is no dose rate effect. So this is the same when you have high dose rate or low dose rate. It's a little bit more complicated when you go to RB for transformation and high LET. In particular, some data rather scary, you see. seem to indicate, like here, that the transformation was enhanced when you go to low dose rate compared to high dose rate. This is, of course, from radiation protection is a big issue, because you assume that low dose rate is reducing the effect, not increasing the effect. So these results with neutrons that are old data were quite scary, and this has never been really confirmed. So this was a big story a few years ago, but then it's been kind of forgotten now. Oxygen effect. Now, one nice thing, one very important thing in the use of particles, especially for therapy, is that the OER is reduced. You see, this is the OER for neutrons, for alpha particles, and when you go to alpha particles, 166 kV per micron, the OER becomes 1. So there is no more oxygen dependence, which again makes sense if you think, because you are now in a situation of direct effect. So the influence of the reactive oxygen species is highly reduced. But you have to go to very high LET. That's something you have to keep in mind. You see here, so the OER goes down with the LET, while the RBE goes up. This is the general trend, and these are actually real data from Berkeley showing the OER going down and the RBE going up. And then, of course, down again when you go to very high LET. These are, but now the problem is this data here on the RBE, these are done generally with 0%, completely anoxic cells, not even hypoxic, but anoxic, which is not realistic. What happens if you change the oxygen concentration? You see, these are experiments now at 0%, 0.15%, 0.5%, 2%, and 20% oxygenation. The OER at 10% survival has a tendency always to go down. You see, according to the approximately same curve, it can be represented by this kind of function, and this is in 3D, the LET dependence, which is here, the PO2 dependence, which is in the different curve, and the OER. So you see the maximum OER of 3, or a little bit lower than 3 actually, is reached when you have low LET and 0%. But when you go into the normoxia region, you see like 1.5, 0.15, 0.5% up to 2%. This is, say, the normoxia region. The OER is actually lower already when you go to these values. But they tend to go to 1. But you see, they tend to go to 1 only at very high LET. So if you want to smash hypoxia with radiation, there is a lot of people working on drugs that reduce the resistance of hypoxic cells. But if you want to smash them with radiation, you have to go to high LET. And these things have been tested also in phantoms, where you can put different oxygen concentration, you can irradiate and measure the survival in the phantoms. We have seen that the main reason for this is that you have a quenching of the free radicals when you have heavy ions, so you don't have any more indirect effect because of the quenching. Now, concerning the cell cycle dependence, what you would expect, I mean, generally speaking, is that you have a very radioresistant S phase. But when you irradiate with heavy ions typically smash radioresistance. No? They have a high RBE when you have a low alpha beta ratio. So S phase typically will have a low alpha beta ratio. So the RBE will be higher here, will be higher in the S phase. So clearly this means that the reduction of the cell cycle dependence is one of the advantages. You see, in this plot, we have a number of advantages in using particles instead of photons for radiotherapy. Some of them are related to the physics. So if you remember the Bragg peak, the fact that you can put the tumor in the Bragg peak, where you have low energy, high LET, high dose compared to those in normal tissue. But now you see the biological one. RBE is higher here than here, because of course of the LET. OER will be high here, but low here, the OER will be reduced. Cell cycle dependence, which is bad, is high in the normal tissue, is bad for cell killing, is good for survival, but will be low in the tumor. And fractionation dependence will be high in the normal tissue and low in the tumor. So you can fractionate without being too much worried, because even the SLD, not only the OER, but also the SLD will be reduced when you have a tumor. So the heavy ion therapy is not only biology, or is not only physics, but it's also biology. Of course, you need high LET. So if you use protons, essentially you only have the physics. But if you go to heavier ions like carbon, you can also exploit the biology. That's the big advantage of heavy ion therapy. Radioprotectors, as I said, this is also something we saw. You would expect that there is no protection at all from radioprotectors when you go to high LET. And going back again to the data of the Hirayama that I showed you in the previous slide, now if you change the LET, and you see it here, the impact is much reduced. These are curves of the of the RBE. But you see what is interesting. The contribution of indirect action, which is about 80% when you are at low LET, should go to 0 here, when you have essentially GOH becomes 0. The G value for the production of the hydroxy radical is 0, but you still have a fraction which is dependent on the indirect effect, showing that maybe it's not only a problem of free radicals, but there is some other chemistry that plays a role even for the direct effect. So you see a 30%, I mean, even at very high LET, you save a 30%, 20%, it's like opposite of X-rays. You still have a dependence from the indirect effect of 20%, 30%, while for X-rays is about 70%, 80%. And the dietary supplements, you see, this does not mean that they don't work for heavy ions, you see, because you see, these are data, for example, for skin cancer. These are animals treated with retinyl acetate here, and you see that there is a reduction of skin cancer also when they are treated with argon ions or iron ions. And concerning the radiosensitizers, of course, also here, not clear whether radiosensitizer like gold nanoparticles work. There is also a matter of cross-section here. You have to hit the nanoparticle. Some data seems to show that there is a 10%, 30% radiosensitization when you use platinum or gold nanoparticles with ion beams. And this concludes this lecture, and we'll be happy to discuss it next time. Thank you very much for your attention.